Hello, this is Sweta Chetankaya, and my research is titled A Genome Analysis to Identify Novel Driver Genes for Pancreatic Adenocarcinoma. First of all, the purpose of this project is to identify novel driver genes in pancreatic cancer through several mathematical genome analyses. My hypothesis was that if the whole exome sequence genomic data for pancreatic cancer are analyzed by using MAF files, or mutation files as we call, and if they're compared with currently known driver genes along with known protein-protein interactions, then the novel driver genes will be identified for PAAD. So what we're actually focusing on is the number of mutations per gene for each sample of pancreatic cancer. This is actually what we're analyzing with mathematics in order to find the novel driver genes. Now, since this is a mostly computer-based research, our materials were pancreatic cancer-specific genome data taken from the Cancer Genome Atlas, which is a free online database. And number 2 was Microsoft Excel. And then I needed 2 softwares, one IGV 2.3 and the other was GI Tools 2.2.3. And lastly, we needed a human protein-protein interactions list, which was taken from the Human Protein Reference Database, which is another free source online. The genome data that was used in the research included 13 files taken from 4 different genome sequencing centers, as you can see in this chart. And each file for each row has different sample sizes. They vary from around 57 to 180. And in the analysis, each file was analyzed separately. Here's the methodology of the research. First, I downloaded the data, the genomic data. Second, I used the sequenced genome data and formed a gene and patient matrix. Then I looked at the high-frequency genes. So these are genes with a large amount of mutations occurring. And after this was protein-protein interactions. And then the next step was a combinatorial analysis with these protein interactions. And after that, we did a Poisson test for significance. And we looked at heat maps. And then after that, I did a network analysis, and then finally comparison with known driver genes, and then I was able to find novel driver genes. So this is a quick overview of the whole process that was completed. As step 0, our first step, we have our data. So this is how each of our 13 files look like. This is a portion of one file, and you can see that there is a row where it says Hugo symbol. So this is actually the gene names listed here. And then it says what chromosome this gene is located in. And you can find other details about it as well. So each row here is one mutation detected within one genome sample that they have. So this gene that you see in the first row might appear several times in one file because there may be several mutations occurring. So each row here means one mutation in this gene. So this is actually information about the mutation that they detected when they're sequencing. So it says this mutation is in this gene, this chromosome, and so on. Now, in order to make use of the files that I had, I made a gene and patient matrix. So what this looks like is you can see here on the very left is a chart where it says genes and number of mutations. So I took this data file that I had and I sorted it out into a matrix where it would give me all the genes that were found to be mutated at least once. And next to it would be how many times it was mutated, the number of mutations. And the next thing I did was I looked at the patient samples. So these are single patients, listed the middle chart here, and it looked at how many total mutations were detected in one sample. Now, the reason I did this is because in the mathematical analysis, I'm going to be focusing on the number of mutations. So that's why from the whole data, all I need is the mutation information. So on the very right here is the mean, standard error, median mode, standard deviation, and etc. You see here that this is the mean value 2.96 of this specific file that I had. So this is just one example of the 13 files I had. And we look at what is the mean number of mutations. So here it is 2.96. What's the median? 2, for example. So this is actually important because I use this in my statistical analyses. So this is just a side note for now. Here is the high frequency step. So what I did here was I used my gene matrix and I ordered all of these genes based on their mutations from highest to least. And this is where I got my high frequency gene list, and I took the top 100 as this list. Alright, so here is where the statistics comes in. As I was mentioning before of the statistical analyses, this is a major method that we're going to use is the Poisson distribution. So what this is it's a method of looking at what's the probability of a rare event occurring. So this is actually similar to binomial distribution, which you may have heard. And in binomial, you have two different outcomes that can occur. And you're wanting to find the probability of a certain event happening. But in Poisson, you don't need to only have two outcomes. You can have infinitely many outcomes. And so this is why we use this for our study, because there can be infinite amount of mutations happening. And an advantage of Poisson over binomial is that we can actually use the mean value of a data set and plug it into the Poisson equation and consider this as the normal occurrence. So when you're trying to find the probability of an event, you're comparing it to the normal standards, which is the mean value. Now you can't do this in binomial, so that's why we're using Poisson. And it's actually you can get more accurate results of probability when you're using Poisson when you have a lot of occurrences rather than with binomial. So if you can see here which equation we used, and in the next slide it'll show a graph of the Poisson distribution. So in the graph here, this is an example of a situation. You can see that there are several hills that are overlapping. So this is actually what's different from binomial as well. In a binomial graph, you have hills that do not overlap, but in Poisson they do. So if you look here, in the first hill we have mu equals 0.1. This means the mean value is 0.1. And then when you increase the mean to 1, you get a more shallow, steep. And then we have mean equals 2 for the third one. It's a lower hill. And then even a lower hill for mu equals 3 and then mu equals 10. So let's say we have an occurrence of 12 and our normal value, so the normal occurrence, which is the mean value, is 10. So this last hill here with the mean equals 10. So if you look here at the 12 on the x-axis and bring it up to see where it meets the hill with mean equals 10, you can see that's going to be the relative expected frequency. So if you have a 10 number of rare events happening, then this is actually going to be 0.1 expected frequency. However, if you had a 12 number of rare events, but your mean value was 3, that means you would have nearly 0 relative frequency happening. So as you see here, the probability of your event happening depends a lot on the mean value or the normal standards. Now that we've went over the Poisson distribution, I can explain how we used it in our research. So what we did here, you can see the chart of our Poisson test. We looked at the mean value of mutations for each file. As I mentioned before in slide 7, our mean value was actually 2.96 for this file. So we plugged in that value into the Poisson formula. And another thing that we plugged in was we did this Poisson formula for each specific gene that we have here on the chart. So we would plug in the number of mutations and then the mean value and find what's the probability of, for example, the first row we're looking at what's the probability of 154 mutations occurring when the normal value, the normal standards, is 2.96. So this is a very big difference. So we can automatically see that there's not a high chance of this occurring. And if you look here, we got 6.7 times 10 to the negative 201. This is nearly 0% chance. So what's significant about this is that even though it has a nearly 0% chance of happening, it still is occurring. And so this tells us that TTN could be a novel driver gene. This could be a gene that's causing the pancreatic cancer to happen. This is actually telling us that this is not just occurring by chance, which is the main key term here. So by utilizing this Poisson test, I took all the values that were less than 1%. And you can even see here, I cropped this, but this is actually a very long line list. And here at 8 mutations and 7, this is actually the borderline where it jumps to 2%. So this is where I can say all genes that have mutations of 8 or above are considered statistically significant genes based on our Poisson test. I had my high frequency genes and my Poisson tested statistically significant genes. I wanted to add a specific aspect to the study, and so I included protein-protein interactions and see how my mutations would collaborate with these. So I downloaded a protein interaction file from an online database, and this is a portion of that file, as you can see here. So in the first row we have protein 1, and the second one we have protein 2. So this means that protein 1 interacts with protein 2, and then it even tells you what type of study actually proves this interaction, as you can see here at description row. So what I did was I looked at how many times each protein occurs in our mutation file. So since these proteins have the same coding as genes, it was easy to locate these genes in my mutation file. So you can see here at count P1 and count P2. So for example, in the first one we have count P1. So ALDH1A1 gene was found to be mutated once in my gene file. And then the second one was also the same. So I did this for each pairs of proteins. And since these are pairs and not just, they don't act in a single position, I wanted to see both of their effects. And I multiplied count P1 with count P2, and I got their weights for each one. After I did this, I ordered my pairs based on their weight from highest to least. And what I did after that is, since previously, in the previous slide, we said that all genes that have 8 mutations or above are statistically significant, I then highlighted all of the proteins here that were 8 or above, and I only took the pairs that had both proteins as statistically significant. So then I called these my statistically significant pairs of proteins. And then I actually conducted a network analysis on this, which I will explain later on. Now, I also included gene size into the study, and the reason why I did this is that when we're talking about chance, we have to think about the size of these genes as well. So imagine you have a large bowl in front of you and a small bowl in front of you. And let's say you're throwing a small ball from a distance, so you have a higher chance of hitting into the larger bowl rather than the smaller bowl. So this is the same thing with mutations. When mutations are occurring, there's a higher chance it will hit larger genes. And so since we don't want these mutations that are occurring by chance, I wanted to eliminate the very large genes that we had. So I looked at their gene sizes, and the ones that were very large, I specifically noted them down, because that could also be a cause of them having a lot of mutations. Here I will introduce a new topic about the graph theory and a network analysis. So first of all, the graph theory was introduced in 1736 when a mathematician, Leonhard Euler, solved the problem of 7 bridges in Königsberg. So you may have heard this, you may have not. So in this diagram you see here, there are 7 bridges labeled 1 to 7, and there are actually 4 different land sections. So this top section here would be A, the bottom would be B, the bottom part would be C, and then there would be D on the right. So A, B, C, D sections, and then 7 bridges. So what you're trying to see is, can you tour the entire city by crossing all the bridges exactly once? Now, this is actually impossible. And Euler actually proved why it is, and I'll explain it in the next slide. So here we have 3 nodes, A, B, C, D, and 7 edges as the bridges that we had. And Euler explains that this is impossible to solve because there are 4 nodes, and if there were 3 nodes or less, then this would be possible. However, it cannot be because there are 4 nodes available. So this is where the beginning of graph theory starts. And this is applied and it's actually extended into several other things, such as looking at what type of clustering occurs within graphs, and not just only small graphs are analyzed, but larger graphs are analyzed as well. So this is what we used in our protein interaction network analysis, as I'll explain later on. When doing my network analysis, I created protein interaction networks as well as gene networks. And in order to analyze these not just visually, but also mathematically, I used graph theory. And by using this, I looked at the node count, which was 255. So this was 255 possible driver genes. So this gene list is our statistically significant gene list. This actually includes our already known driver genes as well. So 255 is our nodes, and then we looked at the clustering coefficient, which looks at how much clustering is going on within your data. And so this varies from 0 to 1, and the fact that we have 0.3 tells us that there is clusterization going on among groups of genes. And then network diameter, this means what is the farthest number of jumps from node to node you can go within a graph. And since the diameter is 4, this tells us that a lot of these genes are interconnected with each other. Since the longest distance is just 4 jumps of genes when you have 255 genes in this network. The next thing we looked at was network centralization, and this looks at how centralized some genes can be to another gene. And so, for example, TP53 is a commonly known tumor suppressor, so this could actually be connected to several others. So when looking at this shows us that since we have 0.23, and this number can be from 0 to 1, there are some genes that are going to be central stations and connected with lots of other genes. So there's going to be a little bit of centralization, also connected to clusterization as well. And then the next one is average path length. And this adds up all the different paths that we can take within a network. So however many jumps we can take and averages that out, which is 1.9. So literally 2 jumps of genes is the average length when you have 255 genes. This is a lot of interconnection. And finally, we have average neighbors and network density. So average neighbors is how many genes that each specific gene is connected to. So that's actually 41.27. This tells us many genes are going to be connected with a lot of other genes. And last is network density, which is 0.16. So this takes the amount of edges that you have and divides it by the possible number of edges. So it looks at, based on the entire possible that could have happened, how much is actually occurring. And this is 0.16. So not everything interacts with everything, is what we're saying here. But of course, since it's not 0, and it's not 1, there is still some clusterization and there's still some interconnectivity, a lot of interconnectivity. So not only did we see this visually, and I will add in the next slides, in the following slides, you will see the networks that I formed. And so not only visually are we going to see this, but numerically looking at this actually satisfies more and assures definitely that there's a lot of interconnection within these genes, which definitely supports the fact that these could be pancreatic cancer genes, since they're all interacting, so they could all be a part of the whole cancer causing list. This is a quick IGV snapshot. This is the software I used to look at the mutations visually, and all you do is upload the data file that you have. And so this was one file, and you can see the entire genome here. All the chromosomes are specified, and then on the top, and then on the left, we have each specific sample that was used. And so if you look here, the blue dots show each specific mutation occurring within that sample. And so at the very bottom, you can see the total number of mutations in the entire sample and their locations. So if you notice, at the very right, towards the right, there's a lot more frequency of these mutations. So this actually creates a very visual look and aspect towards the study. And so this really helped me visualize the whole mutation concept and how frequently these mutations are occurring. This is a zoom into chromosome 11, and you can actually go into specific genes. You can see here, down here at the very bottom, you can see the genes, where they start, where they end, and you can notice the different sizes as well. As we talked about the gene size, that matters a lot too. And so all of these dots here show the mutations, and look, there's a lot of mutations towards the middle where there's SF1 gene, CD5 gene, and so on. So this creates another visual look towards the study as well. And then this last snapshot is a zoom in to a specific section of the chromosome 11. And this is actually useful when you want to look at specific mutations. For example, if you want to see one mutation in a specific gene, you would want to see a better zoom into that. Now, if you click on the mutations in the previous slide, it actually gives you information on that specific mutation. you want to look at. So it tells you different, what type of mutation it is, where it's located, and so on. So here you can see a heat map that was generated by the same software. And so what this does is it technically compares my data of mutations to its own data set of already known driver genes for pancreatic cancer. So here I have around 20-ish driver genes that were found in my own data set. So this really establishes credibility of my whole methodology. Since I found already known drivers as well, the novel drivers that I found could actually be more credible. And if you look here, there's darker and lighter colors. So this just means the darkers are more significant and the lighters are less significant. And so all these boxes here show the different mutations in each sample and in each gene. This is actually the protein interaction network that I was talking about earlier. So you see here that there's a huge map here, and all these blue blocks are different proteins. And since these are protein versions and not the gene versions, in the next slide you'll see a gene network. There are some groupings down here. And there's not just one exact group, there's a big main group which shows a lot of interconnectedness, but there's also small groups on the side. So if we actually looked at this for the different protein interactions to see how interconnected these were, but the graph theory was actually conducted on the gene network in the next slide. Here you can see the gene network, and notice that in the previous protein network we had groups, but in the gene network there's only one single group. This shows us that there's a strong interconnectedness. And as we previously looked at the graph analysis, not just visually but the mathematics also shows us the interactions. So this gives a good idea of how these interactions could be leading to pancreatic cancer as well. Of course, further studies should be done, but it gives a good grasp and visual aspect to it. In the end, after analyzing around 13,000 genes, I was able to find 235 novel driver genes. And among my possible driver gene list, I had very well-known tumor suppressors, TP53, CROSS, SMOD4, and also very recently found driver genes, ROBO1 and SLIT1. These strongly established the credibility of the methodology, so it was a great success for my research. And I'm very satisfied that I was actually able to find 235 novel driver genes. And of course, in order for these to be 100% accurate, they would have to be experimentally tested. But for now, this is a good start to begin with. In fact, here is a small portion of the 235 novel driver gene list that I had. Thank you for watching, and I hope you enjoyed this video.